for years, we've been asking ourselves why myeloma or plasma cells cause osteolytic lesion in one region of the body in a patient, while in other regions you can have subtotal infiltration by plasma cells without any evidence of bone disease. So you can perform a bone marrow biopsy and it comes back positive and 90% are plasma cells, and you don't have any osteolytic lesion over there. But then, for example, you see an osteolytic lesion in the vertebral body of T5 or so. So to analyze this, we used single-cell RNA sequencing and whole-exome sequencing to study the plasma cell compartment. And we also wanted to see whether the surrounding microenvironment somehow contributes to the process of developing osteolytic lesions. So we set up a clinical trial where we basically offered myeloma patients with newly diagnosed and relapsed disease an imaging-guided biopsy of an osteolytic lesion that has been identified by PET-CT. So every patient basically who developed any kind of new lesion in PET-CT was offered to participate in that trial, and then after informed consent, the patient received a biopsy of the osteolytic lesion in CT, and in the same session received a regular bone marrow biopsy. And the good thing is that we were able to implement a workflow where the cells were collected in the morning. So every morning at 7 a.m. we had the procedure. I collected the cells basically and ran over to the lab, isolated the plasma cells, and then we ran single-cell sequencing on the same day to prevent any changes due to a freezing process or delayed sampling, because we have shown in the past that when you perform, for example, gene expression profiling on bank samples that have been shipped for 72 hours or so, you get changes in your transcriptomes. So that we wanted to rule out that by doing everything on the same day. And there are a couple of major findings in our study. And one thing is that based on whole exome sequencing, we did not see that many differences between plasma cells from the osteolytic lesion and the bone marrow. So the mutational profile was very comparable in all of our patients. Basically, we found the same mutations in the osteolytic lesion and the bone marrow, especially in newly diagnosed patients. Only in patients with relapsed disease and extramedullary disease, we found significant differences between mutations called from the lesion versus the bone marrow. But then when we looked at our single-cell RNA sequencing data, and we performed something called an integrated analysis of anchor data sets that have been established by the Satija lab in 2019, where we basically co-register the plasma cells from the bone marrow with the osteolytic lesion. Then you can perform a differential expression analysis on these anchored plasma cells. And then we found in every patient genes that are over and under expressed in the osteolytic lesion. Some of these genes were known candidates, for example, DKK1 and HGF were overexpressed in osteolytic lesions. But for the first time, we saw also downregulation of genes like DASP1 and JUN and FOS that are very important in B cell development. So we identified a couple of new marker genes that we have to study further how they actually contribute into the osteolytic process. But now we were able to, for the first time, to really show that plasma cells from osteolytic lesions are very different from plasma cells in the bone marrow in the same patient. So there's a certain degree of intra-patient and also spatial heterogeneity. And then the part where we studied the immune microenvironment, also very surprising. So we performed a next generation flow to identify 25 different immune cells. And we also performed T cell receptor sequencing on the T cells. And what we found was pretty surprising. We didn't find any differences between the osteolytic lesion and the bone marrow. So from this perspective, we can say that the microenvironment does not I mean, it's a more hen and egg kind of situation. Nobody knows what comes first, but based on our findings, we basically can say that the plasma cells are really different and the microenvironment seems to be pretty much the same. So osteolytic lesion, the driving force is really the plasma cell. When we compared our relapse sample with newly diagnosed disease, it was pretty interesting to see that the T cell richness, so the sheer amount of different T cell receptors that we detected, was very restricted in relapse disease. So you really can see that patients with relapsed myeloma suffer from immunosuppression. And we also analyzed follow-up samples from patients in remission. And what we saw was pretty interesting when we compared it to healthy controls that are publicly available in databases for T cell receptor sequencing. We saw that you basically can restore the T cell richness with successful therapy. So these patients were all in MRD positive CR, one patient in PR, and all patients had the same T cell receptor richness in their bone marrow and the osteolytic lesion compared to healthy individuals. So that was an interesting finding. And then also we looked into the different subsets of T cells that are restricted in relapse disease. And amongst others, we found, for example, Th9 cells to be suppressed. We found more Tregs in relapse disease and less dendritic cells. So you really can see somehow the machinery of antigen presentation and recognition does not work anymore in relapse disease, and you can revert this process by successful therapy. So we are pretty happy with our results, but there's a lot of things that we have to do. Of course, we have to follow up these genes that we found from single-cell RNA-seq, but we are pretty happy that we were able for the first time to show or to gain insights how myeloma cells basically cause these osteolytic lesions in single patients, while in different regions in the body, they don't cause any osteolytic lesions. So these are the major findings of our study.